It's a pleasure to be in this very interesting session. And what I'm going to talk about is about melanins and their potential use in bioelectronics. And I'll give an overview and then some of our work on synthesis, characterization, and then some devices. So bioelectronics is the place where organic electronics and biology meet. And of course, this is a challenging place to do electronics because of the harsh environment. And of special interest is the ionic signals that play a major role in biology and not so much in our current electronics. So this is a place where we have to do some work on finding materials appropriate for that. And one of these materials is melanin. Melanin is a natural pigment which is found from fungus to our cells. It has very interesting properties like a broadband absorption, which is basically the main natural use. It's used to save us from UV radiation, but it has also some other interesting properties which can be used for applications like transistors and sensors and so on. Well, one of the problems of working with melanin is that melanin, from a chemist's point of view, is a very insoluble material, so it's difficult to process if you think about industrialization or trying to make this in a large scale. So we can't do very easily thin films, and this has troubled the community for a long time, but we have found some ways to solve this problem. One of them is using the appropriate pH, but also by functionalization, and I'll tell a little bit more on this last topic. That's what we have been working on this last few years. So melanin basic structure is these two, which we call DHI and DHICA, and then can make many redox forms like the hydroquinone, semiquinone, and then they pack together. Let me put this in the laser mode. And they pack together in quite a complicated way because it's not a very controlled synthesis. So we have many places where there can be some reaction. And in this sense, there is still some trouble in the literature in really saying what is the real structure. But what one knows is that it can form little structures like little graphenes, let's say with a lot of defects, because we cannot forget that we have nitrogen here inside, making it's not a real graphene. Okay. Well, the synthesis in vivo comes from this kind of reaction. We start from tyrosine, and then through oxidations, it becomes this melanin. Okay. And in the case of nature, one important thing to talk about, which we have already discussed in this session, is the formation of hydrogen peroxide, because it can change quite a lot the functions in the end material. And especially what is interesting to know is that it breaks the carboxylic acid. And so you can change the functions in the final material if you change a little bit the synthesis, okay? But in the case of nature, this is done basically enzymatically, where it consumes the H2O2, so we have a predominance of normally of carboxylic acid in natural melanin. So we are not really talking too much about the natural melanin because it's synthesized, and the synthesis is very simple. So what we have to do is we start from L-DOPA and put some ammonia and just do air bubbling. It's a very simple synthesis procedure. Even a physicist like myself can do it. But it has some troubles. One of them is that it lasts 72 hours, okay? So one of the synthesis approaches we have reached for was to try to make this synthesis faster. So we did it under OH pressure in a pressure volume. And then we could make the synthesis much faster, up to 3 hours, okay? And change it a little bit the functions of melanin. The other approach to make soluble melanin was to do it not in water, which is the common solvent found in nature, so in DMSO, and for that we use benzoyl peroxide as the oxidizing agent. And in this case, it takes 57 days. It's even worse than in the case of melanin synthesized in water. But by using pressure, we could reduce this quite significantly. And of course, after the synthesis, we have some steps to do the separation and of the material. Okay. So concerning solubility tests, so we find that we can have the non-functionalized or the hydrogen-made melanin quite insoluble, but with S-melanin, it's better. And you can see it even with your eyes. You don't have to be no special equipment to see that the material precipitates if you put in common solids. Okay. So in the case of NF melanin or water-made melanin, we can make it soluble in milky water. Okay. And in the case of DMSO melanin, we can make it soluble in DMF and P and DMSO. Okay. So concerning the structure, the material made in DMSO is quite similar, apart from, if you look at the FTIR, from this region here where we see the sulfonation of melanin. So we see a functionalization of melanin, okay? And this functionalization is very important. Also, the other interesting thing is that when we do it under pressure, we do modulate the amount of carboxylic acid. And this is a very important group because it can change a lot the way melanin interacts with metals and also with other surfaces. So CO2 is very important. And we can modulate this by using different pressures that can be seen here. And also we can modulate this CO2 in the melanin made in water, which is even more interesting because as we increase the amount of CO2, we are becoming closer to the natural melanin. This is also observed clearly in NMR, and we can see it here that by changing the pressure, we can have quite an increase in these carboxylic acid groups. Okay, so we can see it also in the carbon 13 NMR, and we see an increase in 36%. Okay, and of course that by using a different synthesis route, we get not exactly the same material, which can be interesting if we think about potential applications. I will see some examples in the following. So the melanogenesis under pressure increases the amount of CO2, and the thing is that what we believe is that oxygen is preventing the formation of this hydrogen peroxide, so decreasing the amount of CC cleavage. But as you may have noticed, if we increase too much the pressure, this also decreases the amount of CO2. And probably because of this oxidation of the CO2. So there is an equilibrium of the best pressure that we use. Okay. So this is the overview of the things we have synthesized. So we have NF melanin insoluble. We have soluble in water by using either changing the pH, but better by changing the synthesis under pressure. And in S-melanin, we also can change quite a lot the details of the synthesis by changing pressure or temperature. And this is what we have done this last couple of years. Okay, so let's move on a little bit about what we can do with these materials. And the first interesting thing is, of course, the thin film. This was the primary objective of the initial research. And as you can see, if we look at the AFM and of these materials, and we can see clearly that they are quite different from what concerns the roughness of the films. And we have a best in SML. Okay, so the material done in DMSO is very good at producing very, very nice thin films. Okay. Well, we can see changes of the electrochemistry of these different materials, and I won't go into too much details because of the time. But the nice thing about melanin, it has pseudocapacitive properties, and this can be, in fact, used in applications like we have seen previously, either in pseudocapacitors or in other types of electrochemical batteries and stuff like this. And actually, we have some investigation on that. Well, the other important thing, since we are interested in bioelectronics, is about the biocompatibility. And as you can see here from this very simple test done with cells, that the viability of these cells, they are preserved quite well, independent of the melanin we use. But of course, there are some that are, in fact, quite nice to see. One of them is the S-melanin again, which after 48 hours has a viability of the cells very positive. Okay, so the nice thing about sulfonation is that it has also some side effects concerning biology. One of them is that it has fungicidal effects. Okay, concerning cell adhesion, so we can see clearly also that independent of the melanin we are using, either done in water or as melanin, we have quite good cell adhesion. And so as it's not a surprise that melanin it can be quite good putting inside of the body, since it's already a natural material, even though this is synthetic, okay? Concerning now more the electronic properties, this is the optical properties of this material, and the focus is on S-melanin. And as you can see, we can see here from the absorption against wavelengths, the typical absorption that is on this type of material, where there are no real features. So it's only an increase in the absorption going to the UV, which is basically what I had explained, that the main biological function of melanin is to cutting down UV from our body. But one interesting aspect is that melanin is thought to be a very good material to kill the excited states. So it doesn't have basically no luminescence, but in fact, we could see some luminescence in this S-melanin, which not a very nice quantum efficiency, but it opens up other opportunities for this material in optical applications that are normally just impossible to do with conventional melanin. So we have some luminescence which could be of interest, especially in sensors. Okay, so we have done a more systematic studies on the optical properties, and we have used ellipsometry to see how the behavior of these materials, either in made in water or in the MSO. And you can see here the psi and the refractive index. And so this is very important if you think about using this optically. And actually, what we have seen is that the refractive index is quite stable, independent of the functionalization of melanin. As you have seen, you can change the CO2, sulfonation, and basically from the refractive index point of view, there is not too much of a change. Of course, there is some modulation. Okay. The other important aspect of melanin, and called the attention many years ago, is that it has a free radical signal, which changes, for example, if you are looking in water, not in solution, but in a suspension, from acidic to neutron, this signal changes quite systematically. And what was believed until some years ago, that this signal was coming basically from this kind of state, the semiquinone, which produces a free radical. But as you will see, this is maybe not so simple. So more recently, people were starting to look a little bit more in deep on these properties of melanin, and especially looking in solid state. And what they have found is that actually the signal is composed of two main signals, one which is called carbon center, and the other one which is called semiquinone, even though, as I will show lately, maybe it's not really just semiquinone coming from the signal. So since this is a very poor signal from a experimental point of view, we have used DFT calculations, as our colleague just prior to me was discussing with us. It can be a very nice tool to understand a little bit better materials like melanin, which are very complex. And as you can see, depending on the monomer we use, and we are changing here basically the redox and also the functionalization of these materials, the G factor can change quite dramatically, okay? And so we find out that actually the signals can come not only from semiquinones, but for example, anionic indole-quinone and semiquinone, and the CCR from anionic hydroquinone, and from a defect which is normally not so much discussed in the literature, okay? So we are pursuing this line of approach and have looked in detail in different pHs, for example, and what we can see, in fact, that maybe the signal can be separated into three, one coming from the SFR and two from the CCR, okay? And if we look at, again, looking at the energy levels of these different materials, again, we see, and we like to make a special emphasis on this N defect, which is new, and it comes from the calculations, but it's probably an important defect in this kind of material in general. So as you can see, by using this kind of approach, looking at G values and DFT in combination, we can try to separate these lines and say, for example, there's three lines that were used to simulate the materials are basically coming from semiquinone, but also from hydroquinone and defects and so on. Again, not going too much into details because of time. More recently, we have tried to see this material at very high frequencies, EPR at high frequencies, where we can separate better the G factors. And as you can see, the line changed quite a lot. It has more like a rhombic character. And by doing appropriate fittings, we find that actually there are, again, two paramagnetic species with a G strain, and actually, in the case of high fields, we don't see basically the SFR signal. We only see the CCR, and by following their delta G against rhombicity, we find that we can separate better the contributions to these experiments. Looking now at charge transport, these materials are not very good conductors. And what is interesting is that one is not still sure which is the main charge carrier responsible for the conductivity in N-melanin. There is a discussion on if it's electronic or ionic, and the consensus is coming to a kind of a hybrid material. And this is seen very clearly, for example, if we change the hydration level of the air where the material is sitting on. And as you can see, we can modulate the current quite a lot. Is in the logarithmic scale, okay? So we have some kind of ionic contribution to the conductivity of melanin, and it's clear. In the case of S-melanin, this doping effect or this modulation is not so strong, and in general, the conductivity is lower for S-melanin. And the capacitive effect, we can see also capacitive effect from this hysteresis, okay? So how the signal changes when we go up in voltage or below, which shows us clearly that we have a charge trapping, which is probably coming from the ionic nature of the conduction. Well, as you can see here from the different materials, here is water melanin, Here is water melanin solvent, and here is the DMSO melanin. They are quite different depending on the titration level and the sensibility, okay? So this tells us how the macrostructure is influenced by the synthesis, and that we can also modulate, for example, this ionic character of the conductivity by changing the synthesis, changing the overall material, okay? I'm not, again, going too much in detail, but we can also see this very clearly from the impedance. Okay, now we're looking at the AC response, and as you can see clearly, depending on the hydration level, here is already 100%. We have typical dependence that can be correlated with ionic conductivity, which is very different from the S-melanin, where the electronic character of the conductivity dominates the impedance plots. Okay, so you can see this maybe easier here when we compare directly the NF and the S-melanin concerning the conductivity in as a function of frequency. So we have also tried to do neutron scattering to see the movements of hydrogen, since we believe that ionic conductivity is important in this material. And we have seen effects of drying and wetting the samples. And from the normalized elastic intensity, we can see the quite clear change. Here is the dry hydrogen melanin. Here is hydrated. You can see that the movement of ions is much higher, which indicates that really ionic conductivity plays a role. And for the S-melanin, these values are lower, in good agreement with what we have seen from the transport data. So S-melanin is probably less susceptible to ions. So we have also done some applications of melanin, and one of them is a pH sensor. So in this configuration, what we call is an extended gate field effect transistor. So basically, we have an electrode, and we couple this to the gate of a conventional or for commercial transistor, and we could see quite good responses of this sensor concerning pH. So sensitivity of 48.9 millivolts per pH, which is quite reasonable. And more recently, we have used in combination with people from Swansea in a more sophisticated paper sensor, where not the conventional melanin, not the water melanin, but sulfonated melanin was used. And as we could see, this made quite nice sensors. The sensor responds very quickly to changes in pH, as can be seen in this graph, and they are reproducible. So if we change back to the same pH, we get more or less the same potential. So it's reproducible. It's quite robust, and the potential changed quite a lot. So it's a very, very interesting sensor for pH. It's just a proof of concept. So coming to the end. So I think I have convinced you that we can change or bring melanin to the real world by changing synthesis, and especially by using OH pressure. We increase the solubility of melanin, and also the change of the solubility is coming from basically from what we believe from the amount of COH anchoring the melanin structure. We can also use sulfonated melanin, okay, and by this we have a better biocompatibility, lower ionic conductivity. It's easy to process and can be used in practical use. So, of course, that this work was done with a collaboration of many different researchers. I would like to point out, especially more recently, Paul Meredith and Bernard Mostert from Swansea University. The DFT was done by Augusto Batagin Neto, and we have many other collaborations, Zari Terani and Simon William, which we used to do this final sensor, and Marcelo Munato for the first sensor I showed you. And this is the photo of the group, of course, that with COVID and in Brazil it's sitting quite hard. This photo was taken about 1.5 years ago, and I'm taking especially this photo in separate of João Vitor, who did most of the work I have shown you. He's in the lab for nearly 8 years, working from his scientific initiation to his PhD. And now he has probably will be moving to Canada for a postdoc with Clara Santana. And for those who don't know Unesp and Brazil, so here is Brazil. We are in the state of Sao Paulo, it's shown here. And Unesp is a multi-campus university, so we have campus in 24 different cities. I am coming from here, from Bauru, and it's more or less, if you put in numbers here, 1,900 laboratories, just to give you an idea of numbers. And we are a very strong research university with some strengths in animal science, biotechnology, especially what I'm telling you about is material science. And we have very nice opportunities for the students to work here, and also on theoretical physics, if someone is interested. And very recently, here in Bauru, I have inaugurated a new lab for nanotechnology. So our facilities to do work on this and other subjects is getting better quite soon as COVID is dominated, let's say like that. So thank you very much for your attention.